In this video, we'll work with SPSS to conduct the analyses necessary to evaluate the outcome of the hypothetical within subjects experiment described in the chapter, Design and Analysis of Experimental Research 2. This experiment involves comparing the kinds of flexibility that musicians can achieve after experiencing different types of stretching routines, a control routine, a condition A routine, and a condition B routine. The participants experience all three conditions, thus making it within subject's design, and 10 participants are randomly assigned to the orders specified by a Latin square arrangement, as discussed in the chapter. We're also imagining that we tested the participants following each condition with a flexibility measure that yields a continuous score ranging from 0 to 100. The data file for this example can be found in the file titled Miksza and Elpus within subjects flexibility data. So here we have a subject code indicating each individual. So individual 1's flexibility score in the control condition, their flexibility score after the routine in condition A, and their flexibility score after the routine in condition B, and the particular ordering that they experience. In this case, it's just coded as order 1, order 2, or order 3 as listed in the book. And 0 stands for order 1. 1 stands for order 2, and 2 stands for order 3. So as a first step, we'll want to make sure that any potential effect of the treatment condition does not depend on the order in which the participants experience them. To do that, we'll run a repeated measures Anova that also includes order as an independent variable. So with this statistical analysis, we can look to see if order interacts with treatment condition. In this case, we're hoping for a non-significant result. We don't want the effectiveness of these various types of stretching routines to vary according to whether they had one before the other. So to run the repeated measures Anova, we need to go from our Analyze menu down to the General Linear Model menu into the Repeated Measures option. And SPSS asks you to define what your within subjects factor is called. So let's just call ours treatment to stand for the 3 experimental conditions. And then it asks, well, how many times were these participants measured? And in our case, we have 3 conditions, so there are 3 levels or 3 times. And we add that in. And if you click the highlighted button here, Define, it churns for a moment. And you see there are now 3 spots that are customized to fit the 3 scores we have for each participant under each condition. So we can throw those in there as a group. But we also want to look at order as a between subjects factor, right? There are different people have different orders. Some have order 1, some have order 2, and some have order 3. And by putting it in here simultaneously with our within subjects factors here, SPSS will produce our interaction effect. So we click OK, and in the output menu we have the labeling of the various treatments. So we said we had 3 levels. Level 1 is the control, level 2 is the A, and level 3 is the B. And then it tells us, yeah, there are 3 orders that are coded as 0, 1, and 2, order 1, order 2, and order 3, and there are 10 people in each of those orderings. And then we get a little section called Multivariate Tests. And in some repeated measures instances, it makes sense to think of it as a multivariate analysis. In our case, for what we're dealing with here, you can ignore this box whenever running repeated measures Anova. The next little box you see is the test of sphericity, which is one of the assumptions of within subjects Anova or repeated measures Anova. And actually what we see here is, unfortunately, we have not met the assumption of sphericity because we have a significant Mach-Lees test, which tells us we have to reject the hypothesis that sphericity holds. And so what that means is that when we interpret our results, we have to adjust somehow for that lack of meeting the assumption. And quite honestly, it's rare that you do meet an assumption of sphericity in a repeated measures Anova. That's one of the drawbacks, actually, of the procedure in general. And so here is really the heart of the matter, what we're most interested in. And we have the effect of treatment, a source here for the effect of treatment and the various statistics, the F statistic for the test of that source of variance, and then the p-value or significance level, the interaction effect of treatment by order, and then the error term. And you'll notice here that there are 4 different options for each of these. Well, there are options here that you can use in the case that you do not meet the assumption of sphericity. So we have not met the assumption of sphericity, so we would not report this top line of figures. Instead, we'd report an adjusted line of figures. And a simple move would be to just go down to the next step, and in our case use the greenhouse geyser, and that would work just fine. And you could read further on the various differences between these things, these options here, when you do have violations of assumptions. Some are better for certain circumstances and others for other circumstances, but for our cases here, greenhouse geyser is fine. And what we're really interested in with this analysis is there a significant interaction between treatment and order? And it looks like, if we look at the greenhouse geyser line for treatment and order, no, there is not a significant interaction. So that's good news. Whether our treatment may have an effect or not does not depend on whether they received the conditions in a particular order or another. And then there are more menus that are more options that follow you, and I'll explain those in our next set here in a second. So let's declutter and get rid of this output menu. Now that we've determined that order of presentation is not an issue, we can remove order from our analysis and interpret the result of a simple repeated measures ANOVA to determine whether the participant's flexibility varied as a function of the various structuring routines. So we'll go back into our Analyze menu, back where we were before under General Linear Model and Repeated Measures. We still have 3 levels or within subjects factor there, so we're going to leave that. And let SPSS churn. We're going to leave these in because we still want to compare the participants' flexibility scores across these 3 conditions, but we're just going to take out the between subjects factor. And we're going to add a couple of things now too, since now we're going after our main result. Let's plot our analyses as well so we can get a visualization of the mean differences. And in this case, we're just going to ask for a horizontal view of the different treatment conditions. So add that into plots and click continue. And let's also ask for an estimate of effect size in addition to our statistical significance finding. And fortunately, under this general linear model routine, SPSS will generate those for you. And there's lots of other options, but we need not be concerned with those at the moment. So now let's run this analysis again and take a look at the results. So again, we have our summary. We have 3 levels of our within subjects factor here: control condition, the A condition, and the B condition. We have the multivariate test, which we can ignore. We have our test of sphericity, which again tells us we've not met this assumption. It's less than 0.05, so we cannot reject, or we can reject the null hypothesis that that sphericity is assumed. And so when we're interpreting now our test of within subjects effects, the main test we're interested in the effect of treatment. And we cannot use the top line, sphericity assumed, so we'll skip to the greenhouse geyser line again. And here we have, again, a highly significant finding. Yeah, it seems that we can reject the hypothesis that there is no difference between these 3 conditions. And our eta squared value of 0.54 is actually quite large. So we can say this is a really strong effect. Not only is it statistically significant, but our effect size, our eta squared value of 0.54, is a large effect. Now, SPSS also by default will report a certain kind of follow-up test for within subjects ANOVAs, or repeated measures ANOVAs. You can use those terms synonymously. And those are called contrasts. And in particular, the default contrast that it will give you, and you can set up different contrasts as you dig further into your statistical studies, but the default ones that they'll give you really make the most sense when you're looking at changes over time in terms of repeated measures. So for example, is the change over time a linear change from time point 1 to time point 2 to time point 3? Or is there some curvilinear quadratic change of some kind from time point 1 to time point 2 or time point 3? So that's what these particular types of within subjects contrasts are looking for. And again, since we're not looking at time in this analysis, it doesn't really make sense to worry about these or interpret these results. So interestingly, when we're using SPSS to look at repeated measures ANOVAs, we will often just ignore this default multivariate test it gives us, and we'll ignore the within subjects contrasts unless we've asked for it specifically or want SPSS to give us a sense of changes in trends over time. We'll also get a test of between subjects effects, and again, this is something we can ignore since we're only interested in within subjects differences here across participants. So lots of stuff to ignore when you run a repeated measures ANOVA. To sum up, basically you're interested in how to double check how you've coded your factor, whether you've met the assumption of sphericity, the actual test of the within subjects effect, and that's it. And so then here's our plot here. We have, again, if you remember, treatment 1 is control, and treatment 2 is condition A, and treatment 3 is condition B. And it's pretty clear where the differences lie here. It looks like B is larger than, leads to more flexibility than control. And, it looks like treatment 2 and condition A leads to more flexibility than control. Condition B leads to more flexibility than condition A or control, etc. So although we found a significant effect, that there's a difference among these three levels of the treatment condition, it's still not exactly clear which groups differ from which. We might guess that, yeah, this looks like a statistically significant difference between this point and this point, or between this point and this point. But what about between this point and this point? It's not quite as large. Is that significant as well? So we only know that there is some difference among groups, but we can also run post hoc tests using various methods that SPSS has to offer to determine which groups differ significantly from each other. Or we could even run tests that will allow us to examine every pairwise possibility between these different groups. So let's rerun this analysis and also ask SPSS to do that. So I'm going to retread our ground here, repeated measures. Let SPSS churn. I'm going to leave everything else the way it was, so we're going to still get the plot and we'll still get effect size. But now I'm also going to ask it to look at the main effects of the various treatment conditions. And otherwise, this will give us those pairwise comparisons. And I'm going to ask it to use the Bonferroni correction method so that it controls for what we've described in the book as sort of increased possibilities of family-wise error rate, or type 1 error. So I'll click Continue now and click OK. And so we get the same things repeated again, our coding, multivariate tests that we can ignore, test of the assumption of sphericity, the main within subjects test we're interested in, the contrast we can ignore, the between subjects test we can ignore, and then we get a whole bunch of other menus here where we're looking at pairwise differences between the various treatments, 1, 2, and 3, or control A and B. And then you have significance values for each pairing. And what you can see here is that basically 1 is significantly different from 2, less than 0.05. 1 is significantly different from 3, less than 0.00. 2 is significantly different than 1, and that replicates what we just did. 2 is significantly different than 3, that's a new one, at 0.00. And so basically looking down this column here, any possible pairing of the different treatment conditions are statistically significant. So these follow-up tests, these pairwise comparisons using the Bonferroni correction method allow us now to say that, yeah, there's a significant difference among these three, and in fact, each mean is significantly different from each other.